We all know air pollution is bad for our lungs, but what if it's also harming our brains? Recent scientific investigations are illuminating a critical molecular pathway through which air pollution may contribute to cognitive decline and neurodegenerative diseases like Alzheimer's. This research focuses on a protein called CRTC1 and a chemical modification process known as S-nitrosylation. Imagine something called a snowstorm happening inside your head. Air pollution, from sources like wildfire smoke, vehicle exhaust, and even certain processed foods and pesticides, can increase levels of nitric oxide and related molecules in the brain. This surge can trigger S-nitrosylation, a chemical reaction where a no-related molecule attaches to sulfur atoms within proteins, effectively altering the function of these proteins. CRTC1 is a vital protein for normal brain function, playing a key role in regulating genes essential for learning, memory, and the formation and maintenance of connections between brain cells, what's known as synaptic plasticity. It typically works in conjunction with another important brain protein called CREB. In a healthy brain, CRTC1 and CREB partner to activate genes crucial for these cognitive processes. However, new research with findings published in early 2025 has demonstrated that S-nitrosylation directly affects CRTC1. This chemical modification doesn't prevent CRTC1 from moving to the cell's nucleus, where it needs to be to interact with CREB. But, critically, S-nitrosylation directly interferes with CRTC1's ability to bind to CREB once it's in the nucleus. This disruption of the CRTC1-CREB partnership prevents the activation of genes necessary for maintaining healthy brain cell connections and supporting memory. Essentially, the S-nitrosylation of CRTC1 acts as a blockage, hindering the protein's normal function and leading to a cascade of negative effects on brain cells, including an impaired ability to form new connections and, ultimately, cell damage or death. Scientists have observed this S-nitrosylation of CRTC1 and its detrimental effects in various models. In laboratory-grown brain cells, exposure to molecules that cause S-nitrosylation showed that CRTC1 was indeed affected. In mouse models of Alzheimer's disease, high levels of S-nitrosylated CRTC1 were found at early stages. Similar effects were observed in human nerve cells derived from stem cells of Alzheimer's patients. Encouragingly, introducing a modified version of CRTC1 that could not undergo S-nitrosylation into these cells prevented signs of disease, and in mouse models, this modified CRTC1 restored the activation of memory-related genes and improved synaptic plasticity. These findings provide a direct biological link between exposure to air pollutants and the mechanisms underlying cognitive decline and neurodegenerative conditions like Alzheimer's disease. The research strongly suggests that the S-nitrosylation of CRTC1 is a key event that causally contributes to memory loss. This discovery opens new avenues for developing treatments and preventative strategies for brain diseases linked to environmental factors. The fact that blocking the S-nitrosylation of CRTC1 can improve brain function suggests that targeting this chemical process is a promising therapeutic strategy. Researchers are now exploring the development of drugs that could specifically prevent S-nitrosylation or reverse its effects, potentially offering new ways to protect the brain from the harmful impacts of air pollution. This could lead to new drugs to better treat conditions like Alzheimer's disease by rescuing molecular pathways involved in memory formation. Furthermore, these findings may also help explain why the risk of Alzheimer's disease increases with age, as aging is associated with increased inflammation and nitric oxide levels in the brain, making proteins more susceptible to harmful S-nitrosylation reactions.